I'm actually going to talk about slow fast analysis in the spirit of this session of a model that we have been developing for a while now in collaboration with experimentalists from the University of Bristol, Helen Kennedy, and Daniele Vitabile has contributed to the modeling as well over the years. He recently moved from Nottingham to Amsterdam, and the analysis has been done by one of my now former PhD student, Haron. So I'd like to start with introducing the biological system and the modeling first, and then in the last part of my talk, I will describe the analysis and how far we have got with this. Okay, changing slide. Okay, so the inner hair cells are located in the organ of Corti in the inner ear, and they respond to mechanical vibrations that are evoked by sound waves entering the ear, and have these characteristics hairs on the surface of the cells that are deflected when the dictorial membrane vibrates and depolarize the cells. So they're excitable sensory cells, and they transmit these mechanical vibrations due to sound into electrical signals that are passed down the auditory nerves to the brain and participate the way we are hearing sounds. So during development, these cells fire action cells, which are the main interest of our modeling and have been the main interest behind and motivation behind the modeling. I will introduce these action potentials that have been recorded in mature hair cells. Just want to put their significance a little bit into context. During development of these cells, which in mammals happens in the first week to 10 days after birth, these experiments that I'm showing occurred in mice, and mice are born deaf. So there is a reorganization of ion channels occurring in this period, as well as synaptic maturation, and this is all associated with change in transmitter release mediated by intercellular calcium signaling. This is all believed to participate in the setting of the tonotopic maps in the brain, which then define our ability to hear. So the action potentials in the inner hair cells are calcium-based, and they basically they arise due to the interaction between calcium channels and potassium channels and calcium-sensitive potassium channels via the release of calcium from intercellular stores following influx of calcium from the extracellular environment. So this interaction is well known to result in spikes or action potentials, not in this system, in many other excitable cells, including neurons. The interaction between these ion currents is sufficient to produce a spiking. What is interesting in the case of inner hair cells is that these spikes come in different, this oscillatory behavior or these responses come in different shapes and forms. Here you can see they can be in the form of single spike, they can be bursting, which is characteristic in many cases of a phenomena that have been termed Pseudo-Plateau bursting, where we have a depolarized plateau characterized by small little spikes on riding on it. And there have also been recorded mixtures of spikes and bursts, as seen here in the middle, where in this particular case, the notation that we adopt to denote these behaviors is 3+1. 3 corresponds to the small spikes during the burst. So we have a burst and a spike, and 1 is the large spike. So these 3 traces that you see are experimental data that has been in mature hair cells. The significance of these prolonged action potentials or bursts is in the fact that calcium has the ability to spread further into the cell, thus triggering downstream signaling cascades, including gene transcription, that then participate in the maturation of these cells. So this is just here. Is everything all right? Sorry, I'm hearing some sounds. Can you hear me?
Yeah, yeah.
Okay.
I hear you.
So, thanks. So these this is experimental data, again here showing that indeed associated with bursts in the membrane potential, we have much more significant increases in the calcium concentration. And on the top, you can see a confocal scan image where these yellow spots correspond to a single spike, and the large yellow is spot or yellow red spot is associated to the burst shown here. So I did mention that we have been developing a model for these behaviors for some time now, and as it's commonly done
so
in
did
these
you
systems.
download the origin file?
Oh, no, sorry.
I sent it last night.
Oh,
If you're not the speaker, please mute your sound.
So many people want to talk to you.
Yeah, sorry about that.
Or it's just me.
this is you. This is you. Sometimes it behaves well, you know.
Please hold a second. I'm gonna check.
Okay, I've muted. You can go ahead to talk.
Okay,
Sorry.
thank you. Yeah, I'm sorry. So the model that we have been developing is of Hodgkin-Huxley type, and has the regular, form, which usually is adopted in modeling electrical activity. We have equation for voltage that is given by the currents that interact to produce the behavior, the spiking and bursting in the membrane potential. This is coupled to equations that describe the activation and inactivation of the various currents. And in the case of calcium-based action potentials, we have an equation that describes the rate of change of calcium, which is the fourth equation here on the slide, that is given by flux balance equation, where we have the rate of change of calcium given by the fluxes that contribute to the increase in this concentration minus the fluxes that decrease the concentration, usually calcium pumps. This is just the terms that interact and produce are critical for the calcium-based action potential activity, and that were of interest in our modeling. In other words, we were interested to validate this model based on data where these currents and fluxes are being modulated pharmacologically. So before I show you some results of the model validation, I should mention that we also were lucky to have some experimental data, voltage clamp experimental data that can be used to inform these models. And this is here an example of the activation and inactivation for the voltage-gated potassium channels that that was available in the literature when we started developing this model, and we used in order to inform the form of the voltage-gated potassium channels. Here on the bottom, you can see experimental data and model simulations resembling qualitatively the one large plus burst with 3 small spikes behavior. So blocking calcium-sensitive potassium channels in inner hair cells results in progressive depolarization. This is here following application of apomin, which is a blocker of these channels. And you can see that the spikes gradually broaden, leading to characteristic for pseudo-plateau bursting or prolonged action potential behavior. This is, again, something very typical, and it's found in other excitable cells as well. It's been observed in other excitable cells as well. And in the model, we could account for this behavior without much trouble or tweaking parameter values. This is here represented by this complicated bifurcation where we vary the conductance of calcium-sensitive potassium channels, and we find these different types of solutions, which are mixtures of spikes and bursts, as we change this parameter. Basically, if you look from right to left, decreasing this parameter value changes the behavior from single spike, which is this blue line here, into a series of spikes and bursts with progressively increasing plateaus. So that was the first validation of the model that we did. And the second one came from looking at data where calcium-induced calcium release has been blocked by application of ryanodine. This leads, here is the experimental data showing that blocking this, the calcium-induced calcium release leads to increase in the duration of the action potentials and mixture of spiking and bursting as well. Here is the characteristic pseudo-plateau bursting. So in the model, we could look at the effect of this pharmacological intervention by changing the parameter PER, which is a constant in our model. So here is an example of what we find. Again, we find these solutions organized by isoas, or closed curve of periodic, family of periodic solutions, similarly to the case of when we vary calcium-sensitive potassium channel conductance. And this is an example of one of these isoas that we could identify by varying PER, the permeability of the ryanodine channels on the ER. And this is an example where we see a bursting solution with 4 small spikes and a mixture of spikes and bursts, 1 large and 3 small, and they both belong to the same isoa in this 1 parameter bifurcation analysis. And this is characteristics for other types of solutions like that. I'm not showing here all of them. There are isoas that could be computed, and we were then interested in putting this together by continuing the bifurcations that denote the stability, the stable portions of these branches, which are period doubling bifurcations mostly. In some cases, there are saddle node of periodic bifurcations as well. They both can be computed in 2 parameters. And here is the 2-parameter bifurcation diagram where we vary the conductance of the sensitive potassium channels and PR, and we can summarize the behavior of the model following blocking of the calcium-induced calcium release, where we see progressive change from spiking, that's 1 large spike, into mixture of spiking and bursting, and a prolongation of the plateau of the pseudo-plateau bursting solutions, and blocking the calcium sensitive potassium channels here leads to pseudo-plateau bursting solutions with increasing number of spikes. So having developed this model and validated it with experimental data, we next moved on, and that's really the work of my student Harun, trying to look at the dynamic mechanisms that organize these different solutions by applying some of the slow fast analysis techniques that have been developed over the years by many people. The first thing that we wanted to do is to see whether we can reduce this model. Here, this slide shows some kind of really rough typical timescales that one finds these models based on the parameters. And one thing to notice is that this inactivation variable for the potassium, for voltage gated potassium channel evolves on much lower timescale compared to the others. And so we looked into this in a bit more detail, trying to see whether we can preserve the behaviors, these different types of behaviors that are found experimentally, and the 4-dimensional model supports by putting this variable h in steady state in some sense. So we tried h equals h infinity, which didn't really work for us. We lost the mixtures of spiking and bursting. So the other option that we had is to try to somehow average this variable h. And the way we went about averaging it is to look for calculating the mid-range values between the maxima and the minimum of periodic solutions for a big range of parameter values, and then averaging all these averages. That gave this number. This is not the only number that works, but it's one representative number for us to reduce the model and at the same time to preserve the behavior. This is a comparison between the full 4-dimensional model and the reduced 3-dimensional model, where the parameter values, PER and the conductance of the calcium channel are varied within exactly the same ranges in both cases. And we can see that we can obtain qualitatively very similar behavior. So we are able to reproduce complex solutions and pseudo-Plateau bursting solutions, as well as spiking in the reduced 3D model, similarly to the full 4D model. So having reduced it, we then went on doing non-dimensionalization and estimating the timescales in a way that's well documented in the literature. These estimates for the timescales are based on certain assumptions and limits and bounds. So they're not exact, but they are much better than the rough estimates that I showed in the beginning. In any case, what we found here when varying the conductance of the calcium channel, which has not been done previously and is interesting from the point of view of much hair cells, because there is a up and down regulation of these channels happening during this period. And it's not surprising, given that this is a calcium-based electrical activity, and the calcium is coupled directly, the voltage and the calcium equations are coupled. So what we find in what you see in this table is that changing this parameter affects the timescales of V, the estimates for the timescales of V and C for the voltage and calcium. And then that leads to different possibilities in terms of how many slow and how many fast variables there are. So we considered both possibilities. We have a dimensional system. We consider a situation when we have 1 fast and 2 slow variables, and 2 fast and 1 slow variables, and went on separating the timescales and looking at the critical manifolds and various bifurcations found in the different scalings, trying to see whether we can explain these solutions better. And starting with varying the calcium conductance, this is one parameter bifurcation diagram of the 3-dimensional system, and we can, the rest of the parameters are fixed. Here we have a small range of values where there are isolas and there are these mixtures of spiking and bursting, and there is a big range of values of this conductance where there are kind of prolonged action potentials or pseudo. They could be called pseudo-Plateau bursting, especially for the value of this calcium conductance 24, where you can see the small spiking, the small spikes before the plateau is terminated. This slide here is just really to show that for relatively small and intermediate values of the calcium conductance, neither of these 2 types of separating the timescales actually explains well or satisfactory, depends on what one means by satisfactory, but explains completely these types of mixed complex oscillations that we find, especially mixtures of spikes and bursts. So here in the 2 fast, 1 slow situation, we have 1 slow variable, which is calcium, and we have the characteristic Z-shaped curve of equilibria at the bifurcation of the fast subsystem, and we have subcritical Hopf, and the green surface represents unstable periodic solutions. This is very typical for Plateau bursting, superimposing the trajectory of the full system onto that bifurcation diagram is again very typical and have been seen in many other systems. Shows some some of the typical previously observed behaviors. However, it does not completely explain them. The surface here, the light blue surface is the calcium nucline in this representation. The one fast, two slow scaling, again, which is typical and have been seen in other systems, produces this surface of the critical manifold, and that is parameterized by C and N, which are variables now. And we have a attracting sheet and repelling sheet in the middle, and we have the fold curves, the lower and upper, and we have folded node, but they don't seem to take part in organizing these trajectories either. When we increase, when GCA is increased, the separation of timescales becomes much more pronounced. And I'm here showing again the two types of analysis, the two types of scalings, just for completeness. Have to point out that the here on the two fast, one slow scaling is what is shown is only the case of GCA 24, where we have, if we look at the bifurcation of the fast subsystem as function of C, we see the typical kind of slow passage through Hop bifurcation, and following of the lower branch of equilibria, stable equilibria, and crossing the fold and the null line, and being attracted to the upper branch, following it until the Hop bifurcation, and then passing through it and dropping down, which again is very typical in this case. But if we then look at the situation of fast, one fast, two slow, we can identify specific bifurcations and objects on that critical manifold that are responsible for this behavior in much more tangible and clear way. Basically, what we have here is folded node again on the upper fold curve, and on the middle repelling sheet there is a saddle node, which in the full system, in the case of GCA 24, is a saddle focus that clearly here in the small inset panel C, you can see is associated with the small oscillations before trajectory leaving the unstable or repelling sheet, middle sheet, and dropping down to the lower attracting sheet, SA, where it follows the, in this case, it follows the weak attracting, the weak stable manifold of the folded node. And this same is the situation of GCA 22. Instead of saddle focus in the full system, we have a saddle, and the trajectory crosses the folded node on the upper curve, and then hits that saddle node and leaves the repelling sheet. So finally, I'm not sure how much time I have left.
About 3 minutes.
Okay, I'll finish this quite quickly. Okay, so so we also varied another parameter which just affects the time scale of one of the variables in the system, and this is FC, which is part of the calcium equation, and is the fraction of calcium free to bulk calcium in the system. Here you can see examples of model solutions that we obtained by varying F from a small value to its 1 order of magnitude variation, and that 1 order of magnitude moves us from pseudo-plateau bursting all the way to mixed mode type kind of oscillations through mixtures of spiking and bursting and complex solutions. So in the case of the smallest value of FC that we considered, we have this clear separation of given by 2 fast and 1 slow, and we can see here the bifurcation diagrams of the fast subsystem, the subcritical Hop, and the branch of unstable, initially periodic orbits that then gain stability in saddle node of periodics. And the trajectory of the full system follows these invariant objects in the limit of the timescale separation. So this is just quickly to go through this. The point of this is just to show that if as FC increases, the structure of this, and looking at 2 fast, 1 slow scaling, the structure of this fast subsystem bifurcation diagram doesn't change, but explains less and less well the behavior of the model. And in the case of the largest value of FC that we considered, and the mixed mode oscillations, we have scaling, which is much more better represented by 1 fast and 2 slow. And similarly to the case that I just described, we have a critical manifold, which, and bifurcations here, which can explain the behavior of these mixed mode oscillations. So so basically in this model, we have an example where we go from pseudo-plateau bursting to mixed mode oscillation by changing just one parameter. That was one of the messages of the paper that we published. And I'd like to finish with the open question. After all this analysis, we are still not able to explain the complex solutions, which are mixtures of spiking and bursting, where the separation of timescales is not so clear, and other extended the analysis, the slow fast analysis definitely needs to be extended in some suitable way in order to be able to explain these types of behavior. Thank you very much.
Thank you. Okay, so now if you have any question, you can type them in the chat or pin me in the chat.
I have a question.
Yeah.
Krasi, to be honest, I'm a little puzzled with this a little bit, because you're talking about FC. FC is the free calcium concentration.
Yes.
Fraction of free calcium concentration in the calcium equation, which actually determines the time constant of calcium. So when you look at FC here, 0.000235, and you talk about 1 slow, 2 fast, but then you go to FC 0.00244, which is higher. And now we're talking about 2 slow, 1 fast. That seems a bit strange.
No. Yes, it is on the first glance, but then it's all relative to the time scale of n. And what happens? I need to probably go back here. So if you look here, these values, as you increase FC, what you do is you're making tau c, the time scale of calcium faster, so it gets closer to tau n. So in some sense, this is now has become closer to tau n, and for tau n, this is only the lower bound. So actually, during the spike, these time scales are dynamic. They change as you go along the trajectory. And I can actually illustrate this here. You can see, this is one of these complex solutions, but it doesn't really matter. You can see tau n, this is the lower bound of 0.005. As you move along the trajectory, in this case, it changes quite dramatically. But what I'm trying to say is that these two tau n and tau c are much closer compared to the case of FC 1 order of magnitude smaller. I know that's counterintuitive, but that makes sense with the numbers. So basically what happens is tau c becomes faster and gets close to tau n, and that's why two slow, 1 fast works here.
So what is the relationship, what's the relationship between FC and tau c, the tau c hat that you have here?
It's quite complicated because it's based on this non-dimensionalization. Tau c is a function. Tau c is determined by expression that has parameters, and it's proportional to FC, but there is another term, well, several terms that are involved in that relationships. Several other constants, basically.
Mm-hmm. Okay, thank you.
But as you increase FC, you make C faster. That's what happens, and then it gets close to N, which you agree, right? Because you increase FC, you don't decrease it to slow it. Yeah.
Okay. So there is also a question from Marina. Marina, can you unmute yourself and ask the question?
Thank you. Hi, Krasimira. That's a pleasure meeting you. I actually was looking forward very much to meet you in California, but next time probably. My question is a two-part question. So question number 1, in your equation for ODE for calcium, there is flux. I was wondering, is it constant, and which process you relate to this flux? In your equation, one of the first.
Yeah, so in the calcium equation, we have a flux that's is given. So the influx of calcium is given by 2 components. One of them is the calcium current, that you convert to flux by using a constant that's given by the physical quantities, including also the size of the cell. And the other part of that flux is given by the release from the intracellular stores via ryanodine receptors. So these fluxes are not constant, they're very dynamic. The one that is due to the calcium current is basically determined by the kinetics of the calcium channels. And the one that is given through the release from the ryanodine receptors, that one is relatively simple. It's just a passive flux where you have a constant that gives the permeability of these channels times the concentration difference between the calcium inside the cell and inside the stores, the intracellular stores.
I see, thank you. And another question. These three different types of electrical behavior, did you have a chance to connect it to some primal function of the ear, or do you consider it just some random change in parameters?
So the way these spikes connects to behavior is by setting the sensitivity of the hearing system, the cochlea, to different vibration frequencies. That is basically there are recordings from experimental papers in the literature that show that different frequencies of these spiking and different durations of these action potentials are found along the cochlea as you go along the spiral. These spiking behaviors change, and then they associate to the way the cortex, the auditory cortex, recognizes these various sounds, because the cochlea is such that as you go along, there are different frequencies of sound that the hearing system can encode or can recognize. And that's the significance of these spikes. Yeah.
Thank you so much.
During development, yeah, I have to mention that this is only valid for the development of hearing. So it's in that first initial period when mammals develop their hearing ability. Thanks.
Okay, thank you.
So my talk is about epilepsy and slow fast dynamics, and it's a collaboration with Julien Modolo, Fabrice Bartholomé, and Fabrice Van Ling. We are all working at Inserm in France. So epilepsy is a complex and dynamic disease affecting 1% of the world population, of which 30% is drug resistant. So the general cause of epileptic discharge are hypersynchronization and imbalanced level of excitation and inhibition. So here we see an example recording of local field potentials from a human. So during this 65 seconds of recording, we see several changes dynamically occurring. So first we have aperiodic epileptic discharge, and then they become periodic. Then we have the fast onset where the activity occurs in low amplitude but with very high frequency. Then we enter into the ictal regime, which is the actual seizure where the patient is completely disconnected. It loses ability and conscience. And then finally, the seizure stops. Well, for the drug-resistant patients, the direct brain stimulation appears as an alternative neurotherapeutic method. But of course, you need to determine or know when to simulate. For instance, after fast onset, the ictal period is inevitable. So that's why there are some hypotheses saying that maybe we should stimulate the system during the pre-ictal regime. So that's why we will be considering this pre-ictal bursting here. So of course, modeling the brain dynamics is a challenge, especially choosing the level, the scale of model. So we can go from microscopic scale to microscopic scale. In the field of epilepsy, the mesoscopic scale models, which are the neural mass models, are widely appreciated. So here we see an example in this diagram on the left-hand side, an example neural mass model. So which models basically the average temporal dynamics of neural subpopulation. Here we have 3 of them. One is the pyramidal cells, which are excitatory. The other one is the PV plus GABAergic interneurons. They are somatic projection. They have somatic projections onto the excitatory cells, and they have fast kinetics. The other ones are the somatostatin plus GABAergic interneurons. They have slow kinetics, but they have dendritic projections. So each subpopulation is described by 2 functions. One is a sigmoid function, which converts the presynaptic potentials received by a certain population to firing rates, and then a function called H function, which converts the firing rates to postsynaptic potentials. So the properties of the neural subpopulation are given by basically these H functions. W here represents the synaptic gain, and tau W represents the synaptic time constants. So the output of those functions, are postsynaptic potentials, are multiplied by some synaptic constants, by some connectivity constants. So they, depending on being an inhibitory or excitatory postsynaptic potential, they can be positive or negative. And then this quantity goes to the sigmoid function of the next population, let's say. So in this talk, as I told you, we will be focusing on this ictal bursting regime, pre-ictal bursting regime. We will decorticate the multiple time scale structure of the model, then finally discuss the optimal stimulation protocols which can abort these pre-ictal bursts. Okay, so the main variables of the model are the postsynaptic potentials. Here we see second order as 4 equations of second order ODEs. So we have postsynaptic potentials of the pyramidal cells here, and postsynaptic potentials for the excitatory inputs, postsynaptic potentials for the SOM plus and PV plus interneurons. Okay, so A, B, and G represents the synaptic gains. Tau A, tau B, and tau G represents the time constants, synaptic time constants, and C, I are the connectivity parameters. And this orange quantity that we see here is basically some of the excitatory and inhibitory postsynaptic potentials entering into the pyramidal cells, and this gives us the LFP, basically recorded by the intracranial electrodes. So if you look at these pre-ictal bursts, in fact, we see some change occurring during the course of 13 seconds. Here on the left-hand side, we are zooming onto these regions. First, we have multiple spikes, fast spikes followed by a slow wave. And for the second type, we have one fast spike followed by a slow wave. And by arranging the model parameters, we can match the number of spikes and their frequency, more or less. Okay, of course, to understand what's going on, we need to write our system in a nice slow fast form. Okay, when we pass from these second order differential equations to first order differential equations, of course, we have an 8-dimensional system. We also kind of non-dimensionalize our quantities by the corresponding time constants, and from Y we pass to V variables. We also scale the time by the fast synaptic time constant, tau G, and also we introduce, well, at the end, we have 2 slow parameters, which are delta This is the ratio over tau G and tau A, and epsilon tau A divided by tau V, and they are both small. After reorganizing a little bit, we rewrite our system in the fast form. So the first two variables, V3 and Y8, are the fast variables. The next four are the slow, and the final two are the super slow ones. Okay. Next time scaling gives us the slow system by taking this delta to the left-hand side of the system. Epsilon is still on the right-hand side. And the next one gives us the super slow system, right? Both delta and epsilon are on the left-hand side of the equations. Now, these three systems, of course, we know that they're equivalent for delta and epsilon not equal to 0, but when we take these quantities to the zero limits, they describe different parts of the dynamics, right? In the fast system, well, yeah, okay. In the fast system, if you take delta to 0, we obtain the fast layer problem, and the equilibrium points of this problem gives us the critical manifold S. In the slow system, taking delta equal to 0 restricts the dynamics onto the critical manifold. And we obtain the slow reduced system, which is still a two times scale system, right? With these very slow variables here at the bottom. If we take epsilon equal to 0, we obtain the slow layer problem with the super slow manifold L, given by those equations. In some sense. Okay, yes. So now we have our favorite objects, and this is how do they look like in space of fast, slow, and the super slow variables. So the critical manifold, in fact, has two almost horizontal planes, let's say. V3 equal to 0 and V3 is equal to more or less 165. Then this critical slow manifold bends between these horizontal planes, horizontal faces, but it doesn't fold. Okay. Whereas the super slow manifold has a more complicated shape. It has parts on the vertical plane of this critical manifold. Then it lands on this lower plane of this critical manifold near V3 equal to 0, and it follows like a nice shape. So we also have two fold points, P1 and P2. In between, this L0 is unstable, and for the rest, it's stable. Okay, so we also see a bursting orbit projected onto, superimposed on this nice object. So the bursting, the fast oscillations occurs in the fast time along the vertical face of this critical manifold. And then they land on this stable part of the L curve and 0 curve with the super slow time, pass near the L2 point, jump in the slow time to this left hand side stable branch of L0, walk a little bit in the slow and super slow time, single arrow, pass close to LP1 fold point, and then they bend and follow the critical manifold S again. Okay, here is another projection, right? And in this case, we see bursting orbits with 3 spikes, time course of the fast variable, slow variable, and the super slow variable. Okay, of course, in this system written in the fast form, if we take our epsilon to 0, we can understand the underlying bifurcation structure
because it does what XPP used to do, which is
of the solutions. Okay, so when epsilon equal to 0, our super slow variable V2 becomes a parameter, and this is the equilibrium points projected onto V0, V2 space plane. So we see that we have the stable branch of this stable equilibrium point and the region of stable limit cycles, and the orbit follows this slowly. The stable equilibrium points jumps to the upper branch of this jet-shaped kind of curve, makes oscillations, fast oscillations, and then it lands on this lower branch of stable equilibrium points again. So a couple of slides ago, we have seen that it's possible to change the number of spikes by changing some parameters. In fact, we discovered that the number of spikes is a function of the excitatory postsynaptic potentials received by the fast inhibitory neurons, PV+. So this quantity, EPSP, is scaled by a parameter C5, which is the connectivity parameter between the pyramidal cells and the PV+ subpopulation. So if we decrease C5, the region where we have this limit cycles, stable limit cycles, kind of shrinks because the second Hopf bifurcation approaches to the LP1 point, and like this we can reduce or increase, if we increase C5, the number of spikes. So here we see, in fact, that there is a gap between the bifurcation diagram of the 6-dimensional system and the orbit, but of course, if we take epsilon small enough, and then the orbit matches better with this bifurcation diagram. Here we see how it projects in this 3x scaled fast and 3x scale system, the original system written in the fast form. And still with the same parameters, if we compute in a similar way the bifurcation diagram of the slow reduced system, which is 2x scale, and project this slow oscillation, of course we don't have any spikes in this system, we see that it matches again very well. So just now we can say that the interaction between the pyramidal cells and the PV+ gives us the fast oscillations, and the interaction between the pyramidal cells and the SOM+, the slow GABAergic interneurons, gives us the slow oscillations. So our aim is to stop this bursting behavior, right? So basically, if we stop these slow oscillations, we will be able to stop the bursting behavior. So next, we will discuss the impact of the perturbations on this spiking bursting solutions, but we will mostly consider the slow reduced system because they are the slow oscillations that govern the bursting behavior, right? Okay. So first we will assume that we are applying some, we will be applying some constant input to the system before the sigmoid functions. Here, because basically the input increases the firing rate, so this is the reason of applying such an input before the sigmoids. On the left-hand side, we see the corresponding null surfaces of the slow reduced system. Okay, and in this example, in this case, we are applying this constant input only to the slow GABAergic interneurons. So when we do it, we shift the null surface of this slow GABAergic interneurons. So the black one is without any inputs. So here we have the solution of the full system with loss of spikes. If we apply a negative constant input to the system, we would be shifting this null surface and encourage spiking behavior of the burst. So we have loss of spikes, also fast spike during a bursting. But if we were applying some positive inputs, then we will be able to introduce an equilibrium point on this L0 curve, which was the slow, kind of the slow, super slow critical manifold, or anyway, super slow critical manifold, and that's how we can stop this oscillating behavior. If this input was on the pyramidal cells, we will be shifting the other null surfaces, and positive inputs would encourage spiking, will encourage bursting, whereas the negative inputs, so increasing the inhibition on the pyramidal cells, will decrease kind of the bursting behavior by decreasing its frequency, and kind of decreasing the amplitude of these fast oscillations as well. If two populations, pyramidal cells and some plus were receiving some input, we would have less spikes during the bursts. If the inputs on these slow GABAergic interneurons were stronger than the pyramidal cells, we would be able to introduce a nice equilibrium point and stop these oscillations. Okay, but in a real setting, you cannot apply constant inputs with the electrodes into the brain, because it creates a lot of charge in this cortical area. So that's why experimentally they apply biphasic balanced pulse inputs with these deep electrodes. So here we consider a relatively realistic setting. Biphasic balanced pulse inputs where the pulse width is 0.5 millisecond, and we are changing the frequency and the amplitude of this input. Okay, so the blue area, the blue area represents the loss of bursting behavior, whereas the red ones represents that there are loss of bursts in the system. So in panel A, in fact, we are only perturbing the slow GABAergic interneurons with this biphasic input. So, well, this is possible experimentally in optogenetically modified animals. It's not possible in humans, but we can see that with relatively small frequency, we can abort the bursting behavior and take the system back to a physiological activity. Whereas if we were applying, if all the neural subpopulation were receiving the same input, then we need to increase the frequency of the input in order to first reduce this epileptic discharge and then finally to abolish them. Yes, I think I'm almost done. So basically in this study, we consider complex projectile patterns of local field potentials recorded in humans, and we modeled them by using a very generic neural mass model of brain activity. The slow fast formulation of this model and the 3 times scale structure allowed us to understand and underline the role of the slow GABAergic interneurons in the bursting regimes. And we have also seen that optimal stimulation, in fact, should target these subpopulations, and our predictions support middle to high frequency stimulation with more impact on the GABAergic interneurons. And those predictions are not yet, but clinically testable because they are in the safe regimes. So finally, so this model that we have considered, although initially it was introduced for the CI1 region of the hippocampus, these 3 subpopulations exist in every cortical area. So that's why this model is quite generic. So this is something that we need to keep in mind. So there are some reports experimentally saying that GABAergic interneural activity is enhanced during the epileptic spikes. So this is what we have observed, the increasing postsynaptic potential of the GABAergic mechanisms. And also it was reported, and that's the optogenetic stimulation of GABAergic mechanism reduces the projectile discharge. So this agrees with our predictions as well. And finally, so the moment where you apply stimulation is quite important. For instance, it's not recommended to apply in stimulation to the fast GABAergic interneurons during the projectile phase, because it was observed that such a stimulation increases and enhances the projectile bursting. So this is one of the predictions we made, because basically by stimulation, you increase the excitatory postsynaptic potentials on the GABAergic interneurons and increase the number of spikes during a projectile bursting. Okay, so thanks for your attention.
Okay, thank you, Elif, for a great talk. And we can take question now. So Yami has a question. Can you unmute yourself and ask the question? Okay, so I'll ask the question for you. Question from Yangmin. Has this work been published? And if not, is there any archive version? I couldn't find anything through Google.
So we recently submitted, and we didn't apply. Sorry, we didn't upload the preprint to archive. But if all my collaborators agree, we can do it. So I will let you know. Thank you.
John has a question. Can you unmute yourself and ask the question?
Hi, Elif. I have two short questions. One is, like in terms of the dynamics, what was the role of the SOM to PV projection? And the other is, when you do the biphasic stimulation, you get sort of similar results to if you just applied the positive constant stimulation to the SOM. So I'm curious if you understand why biphasic sort of ends up being the same as positive.
Yeah, okay. So the first thing is to answer your question. So this SOM plus interneurons basically give a negative input here on the pyramidal cells.
No, on the PV is my question. You have SOM to PV.
Ah, yeah. So yeah, they also, so both PV and SOM projects, well, they put inhibitory inputs, but the SOM also projects in onto the PV plus. Yeah, I think this here is the equation. So it goes into this, the postsynaptic potential goes into the equation, goes into this sigmoid function.
So does that end up playing a role in the dynamics?
Is that playing a role in the dynamics? Well, yes. For instance, it has also, this input also changed the number of spikes. And yeah, I think so. If this input was stronger, then the number of you would get less number of spikes during the burst. So this is the kind of thing that I can say.
Okay.
Dynamics, well, I don't know. Anyway, and so the second thing is that, so this biphasic stimulation, I will show you a short slide. Okay, so this is what happens when you apply this biphasic stimulation to the sigmoid function. Okay, so basically, if the time constant of the subpopulation is slow enough, let's say, if the time constant is large, so the subpopulation is slow, then by biphasic stimulation, it's possible to shift this output, so the outputs as the postsynaptic potentials, to a certain level. So this is the blue curve that represents it. So that's why you keep the postsynaptic potentials around a certain level, around an average. So this is how you can in fact stop the oscillations and take the system near the fold of the slow critical manifold here. Basically, you can take the system near this region, because you can well basically stop the oscillating behavior by shifting this constant input. Well, biphasic, but the effect is like a constant, slowly oscillating system.
Okay, thanks.
So today what I want to talk about is a work that we did for the past year with Evelyn Sanders from George Mason University and my postdoc Jonathan Jacquet at Brandeis, who is now leaving to go to Boston University. And the whole work was based on an analysis of the Rule-Cuff map and the link between the Rule-Cuff map, the chaos, and kind of homeostasis. And so before I go into the detail of the model, I want to tell you a bit about homeostasis and a bit of history about it. So the first works on homeostasis were done by Claude Bernard, whose statue was in front of my previous institute, Collège de France in Paris. And he actually found out about homeostasis by doing an experiment on the liver. So it's called expérience du foie lavé in French. So it's a flush liver. And he was actually considering the glucose, the blood glucose that there was in the liver. And he actually washed a liver and found out that the regular physiological levels of glucose were restored naturally by a liver even cut and outside of the body. And so he concluded that the liver itself is able to regulate the glucose independent of any blood or any other system. And he said that the stability of the interior domain, that is called the milieu intérieur in French, is a condition for free and independent life, which is a bit of a strong statement maybe. But homeostasis has become kind of a very important, a central concept in biology. And so basically what we mean by homeostasis is the ability of the system to regulate. So here is the definition from Wikipedia. So homeostasis is a state of a steady internal physical or chemical condition maintained by living systems. And so what I want to underline here is steady. And many systems, I mean, many homeostatic system has been studied since then. As an example, the regulation of the body temperature or the fluid balance or ionic concentrations. And kind of the basic understanding we have about homeostasis is that variables are maintained constant, more or less constant in the body. And chaos is essentially the opposite, I would say. So chaos are systems that kind of, so again, this definition that I put on top is from Wikipedia. They are predictable, chaotic systems are predictable for a while and then appear random. So chaos, I guess a lot of people in this audience know what it is. So it's non-periodic solutions of deterministic systems that are quite complex and that are characterized by disorder and irregularities. And mathematically, when we want to define chaos, well, that's a difficulty. And not because chaos is not a single concept, but mostly because chaos has multiple manifestations. And so there is a paper describing why we have multiple definitions and what are these multiple facets of chaos by Evelyn Sanders and York, 2015 in International Journal of Reflection of Chaos. But a few common properties of chaotic systems are sensitivity to initial conditions. So on the right is a simulation of the Lorentz equation with actually two solutions with slightly different initial conditions. So here you don't really see the difference between the red, the purple, and the blue. But as time evolves, you can see that the trajectories, obviously, they go away from each other and they then become very different. So that's what we call sensitivity to initial condition. Chaos is also associated with non-periodic solutions. It's topologically mixing. It has a positive topological entropy. It's transitive and it can have dense sets of periodic orbits. It can be associated with homoclinic orbit, horseshoes, etc. And there is a conjecture that maybe all these definitions of chaos may not be so different, but they all represent different aspects of the system. All right, so now that I defined the two terms in my title, I want to come to a more recent study of homeostasis done by Yves Marder at Brandeis University and others. That has to do with the regulation of the rhythms in the crab somatogastric nervous system. So the crab has a ganglion, which is a somatogastric ganglion, STG, that has two networks in it, at least two central pattern generator networks in it, the pyloric mill and the gastric mills. And both CPGs are responsible for supporting digestion. And for instance, the pyloric central pattern generator controls the muscles that contract the stomach or dilate the stomach, and it has a three-phasic rhythm. And the gastric CPG, it controls chewing by ossicles, also inside the stomach, and it's a six-phase rhythm. So what I'm going to talk more about is a pyloric rhythm. It's a very simple network. I mean, altogether, both pyloric and gastric mill network, they total 30 cells. And for the pyloric rhythm, it's essentially generated by 12 cells that include, so here you have actually all 12 cells. So A, B are bursters. You have two PD cells that connect to, so you have essentially three populations. AB is a burster that is connected electrically to two cells, two PD cells. And then you have inhibitory connections between all three populations, and they generate bursting rhythms, as you can see in panel A, which is an actual recording of the activity of these populations of cells. Okay, and so what seems to be important here is the order in which the cells are bursting. So PDLPPY and then PDLPPY again. And also what seems to be important is the duty cycle, which is the relative duration during which each cell is active relative to the period of the oscillation. And so what you can see in this figure from Teng and collaborators from the Marder lab again is that when you change temperature, you do change a lot the pattern, the firing pattern of the network, going from slow bursting to quite fast as you increase temperature. But when you rescale the traces according to the period, what you obtain is actually traces that are relatively similar, as we can see in panel C. And I don't know why I don't have my arrow. Okay, so panel C shows a rescaled version of the top figure for 3 temperatures, and we can see that it's very stable. And actually also the duty cycle that you can see here in panel D are conserved when you change temperature. So the fraction during which PD cells are off, LPR on, LPR off, all of that do not seem to vary with respect to temperature. So here in this system, we're talking about homeostasis although the dynamics, as you can see, the neuron do not seem to be so steady or so periodic. But this homeostasis actually breaks down if you increase temperature beyond 23 degrees or 30 degrees. And so these are the breakdown that you see. You can see that if temperature is too high, then you have cells stop firing first, and then they have kind of irregular activity. It's really bad that I don't have mine. Okay, so I'm gonna show you like that. So when temperature is very high, you don't have spiking, and then you see appearing when temperature decreases, irregular activity that are chaotic in the sense that we are used to. So this is completely irregular. And as temperature decreases even further, at about 22 degrees, then you recover regularity as you want. So the animal is still able to digest. And so in a sense, we have good chaos, which is a chaos associated to the rhythms, because I mean, models of this system are chaotic bursters essentially, but they are able to maintain constant duty cycles and the order in which you spike. But when you increase temperature, then you can break down these rhythms. And here are two examples of a model also proposed by Yves Marder, where the rhythm breaks down and also the order breaks down and the duty cycle is broken. And from the mathematical viewpoint, what we see in both models is that it's chaos. But there is a sense of good chaos where the crab is able to digest, and there is bad chaos where the crab is essentially burning. And so that, is there a question?
I don't think so.
Okay. Okay, and because it's a mini symposium on slow fast systems, I'm going to call the good chaos a fast chaos and the bad chaos a slow chaos. I mean, for other reasons. But to study this question, we actually came studying a simple bursting neuron, one of the most canonical models of bursters, which is a discrete time model, which is given by the Rukov map. The Rukov map has a nonlinearity in the fast system. So X is a fast variable. And it has this nonlinearity, which is alpha over 1 plus X squared. And the slow variable is just a simple linear dynamics. Right, and so when you change parameter, I mean, the system is very well known. When you change parameter, the parameter alpha is kind of a key to the system. When you change parameter alpha, you go from a fixed point to a periodic orbit through an Emax-Acker bifurcation, actually. And then you start having bursting. When you start having chaos in the chaos, like this bursting become chaotic. And then you can start having this kind of shortcuts that you can see that are kind of disordering the dynamics. And then the dynamics becomes completely disordered as you keep increasing alpha, and especially beyond the value alpha equals 4, but I'm going to come back to in a short while. So that's the classical dynamics for the Rukov map. And it's also well known that the Rukov map is a slow passage through a period doubling cascade. The fast variable xn has a period doubling cascade that I'm going to show you as you vary alpha. And the slow variable is actually very simple. It's going down when x is larger than sigma, and it's going up when x is less than sigma. Okay. So what's important to understand here is how changes the fast dynamics as a function of alpha. And so here what you can see overlapped are the kind of the null lines that are the kind of the equilibria for the fast system. And overlap to it, you can see the attractors of the fast system. So 10,000 points for a simulation of the fast system for a fixed value of y. Okay, so x is my x-axis and y is my y-axis. So you can see that you have bifurcations, and then you have chaos arising with this period doubling cascade forward and backward. And then this kind of strange attractor grows, it merges into one attractor that inflates, touches at alpha equals 4, it touches actually the unstable fixed point, And at this point, you have what is called a crisis in discrete dynamical systems. And so the shape of the strange attractor is completely modified when the strange attractor touches this unstable fixed point. So it's essentially disappearing. And the trajectory is actually, instead of having chaos, they just go to the fixed point that is on the left, right? All right, so that's essentially the dynamics that the Rule-Curve map has. And we then ask the question, when is the Rule-Curve map chaotic? And so for that, we use the technique which is based on the Birkhoff ergodic theorem. So we used weighted Birkhoff averages. That it's a technique that has been studied by Evelyn Sanders and York and collaborators and others also. And so what you see here, B of H, is the Birkhoff average of along an orbit of the dynamical system. And this B of H converges when the system is ergodic, when F and H are smooth. And the speed of convergence can give you a hint of whether the trajectory is regular or chaotic. So actually, if the orbit is regular, then the convergence of the Birkhoff average is of order 1 over n. But if the orbit is chaotic, then the convergence is order 1 over square root of n. And basically, the proof is kind of a central limit theorem. And so what we did was compute the Birkhoff average and matching digits on the Birkhoff average. And so a high value means that the Birkhoff average has converged very close to the limit, and a lower value means that you have less matching digits. So it's essentially the log of the error. And so that means that you are probably chaotic. So we have computed these Birkhoff averages, these matching digits for the Birkhoff average, and we also confirmed whether the trajectory was a periodic orbit or not by using a Newton method. Okay, so in blue are so in green are the orbits that are found to be a periodic orbit. Okay, and so we can see that we have the emergence of a chaos with a bistability region, which is associated actually to the Devil's staircase that there is in the number of rotation of the system. And we can see that we have chaos arising way before 4 at about 2.7. And for these values, it's unclear that the system is chaotic. So when you look at the trajectory, this is what you see. So you see that the dynamics, I'm gonna stop making my screen very large because I cannot see, I cannot have my pointer. So this is a trajectory that you see that really looks like something completely regular. I mean, completely quasi-periodic, and the dynamics seem regular, but it's found chaotic by this criterion. So then when we look very closely to this trajectory, we actually found indeed that there was a horseshoe somewhere. And so we call that a weak chaos or an invisible chaos. And it seems that all mathematical definitions for chaos say this is chaotic as much as anything else. But however, it's somewhat less chaotic. I'm gonna have to go faster, actually, much faster. So, so here is what happens when you have for the dynamics overlapped with the attractor for 2 different values of alpha. So for before alpha equals 4, what we can see is that the bursting is irregular, like chaotic. But if you blink, if you don't look at the details of the bursting, then it's kind of regular. First, the rhythm is maintained, and then the duration of the burst is maintained, and more or less everything is maintained except the precise timing of the burst and the precise height of the burst. So what happens in terms of dynamics is that we transition along a fixed point through a strange attractor, like in this diagram here. And this switching between the two is kind of regular. And then the timing of the switching is kind of controlled by the slow dynamics, which is also regular. And so what you see is indeed that you have chaos in the system, but this chaos is not affecting the overall structure. Okay, so this is what we call fast chaos. The fast dynamics is chaotic, but the slow dynamics is essentially regular. So this happens in this situation, but also when you have the crisis and when you are sure to transition here after the crisis. Okay, so in that case as well, you're going to have fast chaos. Now, when you have the crisis here and disappearance of the attractor, then you have, then we can see that the trajectory either takes a shortcut and comes back. So these are short bursting trajectories like these ones here, or they go all the way through the bottom of the attractor and they do a full burst. And we have an alternation between the two. And so in that case, the chaos that was present in the fast variable is kind of leaking through the slow variable. And so what happens is that through a trajectory, let's say I start from here, I'm going to go up, I'm going to be in the chaotic attractor for a while, but then there is no more chaotic attractor, but there is a ghost of the chaotic attractor, and I'm going to have a transient chaos that is pretty long. But, I mean, this has been estimated by Gregory and other people, and Arnold at the crisis, the duration of the chaotic transient. And so you have a chance actually to take the shortcut and go back here, especially when you're very slow. But when you're not so slow, you have a chance to also be in the chaotic attractor and never end up on the left side of the unstable orbit. And so go all the way back to the chaotic attractor, go down. And so now you have a probability here, a transition probability to go from this side to that side. And so you have two ways to go from B1 to B2, which would be two regions here. One would be through the relaxation oscillation, classical, and one would be through the shortcut. And so now this changes the picture in that now you have, this is not the picture I wanted, but now you have a probability P to go on the other side here, and a probability 1 minus P, sort of, to go all the way down to the attractor and do a full a full relaxation oscillation. And so the probability to switch, we estimated it as the area of the chaotic attractor, of the support of the ghost of the chaotic attractor, and that is on the left of this unstable fixed point. And so with that gives us a probability to switch, and we have been able to count the number of long orbits versus taking the shortcut using this probability that is estimated as an integral here of the mass. And we see that this kind of predicts well the number of small oscillations versus big oscillations. So essentially, let me go real quick because I see it's 11:30, but all right. So what I just wanted to add is that the way we explain this transition from slow chaos to fast chaos does not depend on the fact that we are looking at the Rukov map. It was very handy and it gave us all the intuition. But essentially what we say is that a transition between slow chaos and fast chaos will be associated with the presence of relaxation oscillations with a chaotic fast variable and the presence of an internal crisis in the strange attractor as you change the parameters. And so these phenomena are quite general and they can be found in various models, including, for instance, ODEs and reversible maps. And so we have a few examples here. Here is an example on a cubic map that has been proposed by Han and collaborators. And this is just, so the fast is just a cubic map, right? And we added the slow dynamics on a parameter here that you can see in red. And so you can create with this system. So this system has crises, actually, has internal crises. And so you can see that as you change parameters, you get through the crisis. So here, it's not the right image I wanted. But this is, so you go from fast chaos to actually slow chaos. But this is, the top image is not the right parameters, sorry. And we can also create a modified Lorentz attractor, putting a dynamics on the R parameter, the one that sees a transition from fixed points to chaos through the Hopf bifurcation. So here again, I need my my mouse. So here are the fixed points. You can see that we have a bistability, which is already well known. It's been shown by York. And so this you have here, you have bistability, and then all these colored attractors are strange attractors. And then you have a crisis, which comes from the intersection of the unstable limit cycles with that originate from the Hopf bifurcation with the chaotic attractor. And so once we saw that, I mean, it's easy to generate a relaxation cycle that would take you from the fixed points to the strange attractor, and then when you're on the strange attractor, you go back to the fixed point and come back. And so this has all the elements we want because it can have relaxation cycles and it has a crisis. So in particular, what we don't see is that it has transient chaos here. And when we simulate this, we do obtain fast chaos for some parameters and then slow chaos. So alternations of different types of bursts in for an overchoice of parameter. So, all right, that's all I wanted to present. I skipped over a lot of related works, in particular works from Teresa Che and Rinzel that looked at the model of excitable membrane, I think for pancreatic beta cells, where they do have transitions between regular bursting and irregular bursting all the way to spiking. And these models do not really have fast bursting, chaotic bursting, like fast chaos in the sense that we defined it. And essentially because these models can be also defined in 3D. And so the slow variable, I mean, when you remove the slow variable, your fast dynamics is essentially 2D. And what happens here in this region, although it's a 3D slow fast system, is that you get close to a homoclinic orbit. So this has been studied in detail by Thurman. You get close to a periodic orbit where essentially the separation of timescales is no more true. And so you interact, I mean, the slow dynamics, the fast dynamics become slow because you get closer from a homoclinic orbit. Essentially, this is my takeaway from that, but the paper is extremely, I mean, these two papers are remarkable. There is also a lot of work on the study of bursting in different ODEs and so fast ODEs, and that's essentially all what I wanted to go because I got over time, so sorry for that. So my conclusion is that chaotic system can maintain regularities that are good enough for functions, although mathematically they are defined as chaotic, and a type of system that can support this kind of fast chaos or regular enough chaos include this class of slow fast systems that have a fast chaotic variable and that have relaxation oscillations through these strange attractors. And this approximate regularity that you have in these relaxation oscillations, including chaos, it actually breaks down when the strange attractor actually hits some invariants of the slow dynamics, which are defined as crises in chaos theory. And actually this kind of dynamics can be found both in discrete time dynamical systems and ODEs. And so that's it. Sorry, I was a bit long and rushed at the end, but thank you for your attention. And I want to thank Jonathan Jacquet, who was kind of the inspiration of that, and Evelyn Sanders. All right, thank you very much.
Okay, thank you. Are there any questions? You can type your questions in the chat.
Hi, Jonathan. Can I ask you a quick question, please?
Uh-huh.
How do you find sort of taking together continuous dynamics and discrete dynamics for analysis? Did you encounter some problems?
For the
in general, you're analyzing continuous chaos, but the models, the methods you used, some of them are discrete.
yeah. Yeah, so most of the study, I mean, most of the method is done with a discrete time dynamical system, because this was kind of the simplest, and it was known that it has this leaking. I mean, this was already shown, this kind of shortcut. So we actually started doing all our study with this model, and it's only after finding out what could be the source of this kind of difference between slow and fast chaos that we were looking for exactly a structure that contains what we need, namely a fast chaotic attractor and a slow passage through a crisis. And so that's how we then selected the models. And so the two other models that I showed are a discrete model and one is a continuous model. But generally, whenever you have like a crisis, I think it's not hard to do a relaxation oscillation through it. I mean, it just takes a few tweaking of the parameter. I mean, making the parameter dynamical and then using the hysteresis. But it's actually not so common. I don't know. I mean, I don't know a lot of models that do have this kind of structure.
Okay, thank you.
So let's see.
I have a suggestion. Interesting talk. My chat is disabled, so I can't type for some reasons, right? Some years ago, we published with Christian Mirat, we published a paper on non-reducible maps. So if you happen to go to my website and look at the very bottom, you will see some interesting similarities.
Okay.
I can't give you a link here for some reasons.
All right. Okay. Yeah. I'll have a look. If I don't find, I'll email you.
Oh, sure. Of course. Right. Anytime. Thanks.
Okay, hello everyone. Can I please ask everyone to mute themselves? So that could be easily, because it's very confusing when sounds are around. Okay, so today I'm going to talk about non-sequential spike adding, and thank you Anmar, Jonathan, and Yang for organizing this. This is something good, at least for me, to present my work, the good side of COVID-19, because if it was in the US, I was not able to attend. So it became an opportunity for me to present here. So yes, this work has been done at McGill, and I moved recently to UK at the University of Surrey to start a new job. Okay, so let me see. Okay, so cerebellum is located in the back of head, and it controls the body posture, balance, speech, and so the input that it received from senses like eyes and ears, and then it kind of fine-tuning the muscular movements. So the core output of cerebellum is generated by Purkinje cells. Purkinje cells are inhibited and excited by different sources, in particular stellate cells. Stellate cells themselves are excited by parallel fibers from granule cells and inhibited by stellate cells and other sources like basket cells. So these cells are spontaneously active, and they tonically generate spikes and action potentials. In this way, they are kind of boring to look at, but they have some properties that makes them very exciting and interesting. The first one is that their properties change during the time as you start patch clamping. We call this as run up, and at the moment, at the baseline, when you start patch clamping, we call it pre-run up, and after around half an hour, the cell kind of stabilizes and its properties remains almost unchanged. So at the beginning, for the pre-run-up, the frequency is kind of lower, the threshold is different, the amplitude is different, and during post-run-up, clearly frequency increases and also threshold and amplitude changes. The other property that they have is non-monotonic latency. So what is latency? So first we hyperpolarize the cell, and I should mention this is done by Molyneux et al. Before I took it from there, but what happens? We hyperpolarize the cell to turn off the firing, and then we apply a current beyond the threshold, this dashed line, and then from the moment of applying the current to the top of the first spike is counted as latency. So when you look at different hyperpolarizing currents, you see that the latency in stellate cell at the beginning it increases, and then at some point it decreases and then plateaus. This is in contrast with other types of cell. For example, this is latency in granule cell that monotonically increases as you hyperpolarize the cell. Okay, and the third and last property is that they switch in response when you apply a couple of inputs. So first inhibitory input and then excitatory input. So the way this input is applied is that you hyperpolarize the cell and to again stop firing, and then for 15 milliseconds an inhibitory input is applied as it's shown here on the left, and then an excitatory input follows it. So for different magnitude of inhibitory input, you get different responses. For example, for black curve here, you don't have any kind of inhibitory input and you get a single spike. And then for more inhibitory input, you don't get a response. And finally, when you even increase it, the inhibitory input more, then the cell become responsive. Okay, so now we want to look at this from mathematical point of view and model it. So this is this model developed by John Mitri, and we took this and we look at its properties, and I would go through the properties that it can capture, and just as a review, and then I would go to bursting part. So this is the Hodgkin-Huxley type that Krasi talked about it this morning. So sodium current, delayed rectifier, and leak current. These are typical currents that are included. Also, the model includes A-type potassium current and also T-type calcium current. Okay, so this is a type 1 oscillator, which because of the SNCC bifurcation that exists in the bifurcation diagram with respect to IAP. So because this is important, I just go once through these bifurcations because it's gonna appear quite frequently during the talk. So the solid means stable, and dash means unstable. So there is a stable branch of equilibria here, and then it goes through a saddle node or fold bifurcation and then becomes unstable, and then another saddle node bifurcation and it still remains unstable, and then a hop bifurcation, and then it's becomes stable. So at hop bifurcation, a branch of periodic orbits comes out which are unstable, and they become stable at a saddle node of periodic orbits. And this is where the tonic firing happens. Okay. So the first one is the non-monotonic latency that is similar to experiments has been done, and as you can see, it's very similar to the experimental results. But to explain this mathematically, we apply slow-fast analysis on this. We take the slowest variable of the system, the inactivation of A-type potassium, and treat it as a parameter. And the bifurcation diagram looks like this. So there is a SNCC bifurcation and stable branch and a small region of stable periodic orbit. Okay, so superimposing the HA null line on top of this, it shows that when the cell is hyperpolarized, there are 3 equilibria, 2 is stable and 1 unstable, and E1 is the equilibrium that the cell is gonna be set at when it's hyperpolarized and it's silent. Okay, now applying the eye test to let this system, let the cell to generate a spike and start tonic firing, and there is only 1 equilibrium in the middle, and so the equilibrium E1 is displayed. So here in the inset, you can see that where it's sitting. Now, the black one is black curve shows the location of the fast subsystem when eye test is applied, and the equilibrium is gone, but there is a very narrow region between the 2 null lines. So that creates a bottleneck which causes a slow region, or also it's called ghost of a SNCC, and that causes that non-monotonic latency. Okay, and finally the switching. So we replicate the input into the system, the synaptic input by this equation, and so we were able to compute the boundary between the responsive region and non-responsive region with respect to conductance of inhibitory and excitatory input, and that shows that the boundary looks like an S-shaped curve, and therefore, when you take a fixed excitatory conductance, and you change inhibitory conductance, you can see that the system can switch between response and no response as inhibitory input or the magnitude of inhibitory input increases, and that causes similar switching in the response of the system. Do you see my pointer, by the way?
Yes.
Yes.
Okay, great. Okay, so looking into the system, we identified that in fact the boundary between the responsive and non-responsive is defined by a connecting orbit between the stable equilibrium on the lower branch and the saddle equilibrium on the middle branch, which I showed you before in the fast subsystem. Okay, now I'm gonna switch the gear here and talk about bursting. So our collaborator that I should have mentioned at the very beginning, because this is joint work by with Ryan Alexander, Dirk Bui, and Anmar at McGill. So Ryan recorded this bursting by applying 4AP, which is a is known for a potassium currents blocker. So it significantly blocks A-type potassium, which kind of strange for cerebellar stellate cells. But he looked into this and he realized that, okay, there is no problem with his experiments. And so we decided, okay, so maybe this is real. We decided to reproduce this with our model. And whatever we tried, we were not able to do this, and we failed. So we guessed maybe some currents are missing from the model. And we decided to include some of the currents, some channels which are expressed in the cell into our model. And therefore, we added high voltage activated calcium currents and also calcium activated potassium currents. And I think also Krasi talked about these currents in the morning. And so calcium flux is going to be also necessary for these currents as well. But surprisingly, including these currents also didn't help us, because we were thinking that, okay, 4AP is a blocker, so we have to reduce kind of conductance of the channels, but we failed. But at some point, we realized that in fact, we can generate similar kind of bursting, but counterintuitively by increasing conductance of high voltage activated calcium channels, which is shown here. But yeah, that was a surprise, so we were not convinced. But looking into literature, that we found out that actually there are studies and there is strong evidence that 4AP can even increase conductance of high voltage activated calcium channel by 3-fold. Okay. Okay, so looking into different variations of conductance of HVA, to look at the bifurcation diagram, okay, there are many things going on here, but I just focus on the part that I need. So there is a half bifurcation here. That stable part comes out that this in fact continues here, but I had hard time to compute it. I was not able to do it. And I will come back to this part later. And then there is an isola periodic orbits here, which is stable at this part and then becomes unstable at a period doubling here. And there is a family of isolas as well, which I showed them in different colors. So I zoom here to see how they look like. So this isola that I showed you correspond to tonic firing. And these isolas are associated with bursting. So as you see, each one of them has different shape and the number of spikes. So the numbers here shows the number of spikes in each of these isolas, and the number of spikes for each isola remains fixed. So in this one, there are 11 spikes, and then 12, and then decreases to 10, and then 12, and then very irregular. And the stable part of these isolas from right sometimes is saddle node of periodic orbit, or sometimes is period doubling, and similarly from the left. Okay, and another conductance that we targeted to look at was the calcium activated potassium channels. And similarly, we see a wide range of parameter that there are 2 spikes per burst, and then that comes into a family of isola again that I zoom in again to see that how they look like. So here it seems it's the shape of the isolas and also the order of bifurcations are more regular, also the number of spikes. So on the left, there is a periodoubling that at a periodoubling, the stable part of the isola becomes unstable, and from the right is saddle node, and also the number of spikes, so 3, and then there is no 4 spike isola, and then 5, 6, and then there is no 7, 8, 9, and there is no 10 and 11. But here, still, at least they increase in order. So it's kind of, you can talk about a kind of a global trend for increasing the number of spike when you decrease conductance of calcium activated potassium channels. Okay. But looking into 2-parameter space, it shows much more, at least for the right panel. But on the left, in the left panel, it shows that, in fact, conductance of A-type potassium can change the number of spikes. That's at least the only effect that we can see here in this 2-parameter space. So the red and yellow curves here show the period doubling and saddle node of periodic orbits that I showed you in the previous panel, these saddle node and period doubling that are labeled here. And so there, it's, as it can be seen, there is a small bistability region that separates the region with different number of spikes. And as one parameter changes, some regions disappear like this one, and some other regions appear as they switch their position. Okay, but in the right panel, it's much more is revealed. So in the middle, we have the bursting that we label SW, but we have other kind of activities. So here, for the brown region, we have tonic firing, and for the black region, we have elevated steady states that it converges to the stable steady state on the top branch that I showed you very beginning. And then we have Pseudo-Plateau bursting, which is shown in dark blue, and then gray, which is chaotic bursting, which is kind of lies between tonic firing and square wave bursting. So for the transition to tonic firing, it goes through the chaotic region. Okay, so we want to look to some of these dynamics, how they are generated. So now we have 2 slow variables, and one is HA that we had in our previous model as well, and also calcium. So for a specific calcium concentration, the bifurcation diagram with respect to HA looks like this. Everything looks like similar to what I showed you before, except for these branches of periodic orbit. First, the periodic orbits coming out of the hop bifurcation terminate at the homoclinic, and they are unstable, but there is an isola of periodic orbit here, which small region of it is stable, and then at the tail becomes unstable at a period doubling bifurcation, and then terminates at the homoclinic and one side, and then from the other side also it terminates at another homoclinic. So now putting together this for different calcium concentration, we have a surface. So I removed the half bifurcation that we had here to make it clearer, but putting together all the isola gives us surface of branches of periodic orbits and also surface of equilibria, which is shown in gray. So now, when the periodic orbits drops down to the lower branch, so if I go back to this branch here, it goes to the stable branch of equilibria, follows it all the way to the fold, and then it goes around the fold. So if I come back to this, goes around the fold and hits the stable part of the periodic orbits. And then as it moves, calcium concentration increases, then the stable part of the isola becomes narrower and narrower. And at some point it disappears, and therefore the bursting stops and then drops down. So that can be seen here much easier, because here now I just kind of project down everything into a plane. So in the CA-HA plane, so you can see all those bifurcations, how they look like. So from the fold, it jumps up, start oscillating, and then it reaches the homoclinic and then drops down to the lower branch. And then that cycle repeats. Okay, so as I showed you in the 2-parameter space, that the system is able also to produce Pseudo-Plateau bursting. But the question was, to go back to our collaborator, is the cell able to do so? And the answer is yes. So, but by applying kind of another pharmacological agent, which is cadmium here, and so cadmium blocks high voltage activated calcium channels, which is known as a calcium blocker, but it's also potentiates the calcium activated potassium channels. And this is, in fact, is the same thing that is done in the model to generate the Pseudo-Plateau burst. Okay, so if you want so fast analysis as well, so now in the fast subsystem that is shown here, the isola, which was close to the saddle node SN1, saddle node of equilibria SN1, it's gone, so it doesn't exist anymore, and therefore, so when the periodic orbit jumps up to the upper branch, this stable periodic orbit don't exist, so it comes down to the branch of equilibria and follows it all the way through the hop, and then drops down to the lower branch, and the cycle repeats. You might ask a question that why, after passing through the hop, it stays close to it, and that's the effect of it's the effect of a phenomenon known as delayed hop, which has been studied before. And just to conclude, we know that, for example, for AP, it's known as a dirty drug for experimentalists, and also cadmium, they interact with other channels as well, and the combination of all these channels and their interaction is able to generate such sophisticated dynamics, and the model captures many features that I showed you through this talk, but the model itself is quite rich in dynamics and can generate many behaviors that some of them I just skipped through, and I explained just 2 cases. But the question is that what's the point of having this machinery and these ingredients in the stellate cells in general, and also during developmental process. And yeah, in a specific case, for example, how the chaotic bursting generated, what's the underlying dynamics for chaotic bursts. And final question would be that why this irregularity emerges in the number of spike for the isola, because in the classical slow fast analysis, they've shown that the number of spikes increases in the isolas just by 1, and this is kind of peculiar. And I want to thank everyone for paying attention.
So thanks, Saeed. We just have a very short time before the next talk is supposed to start. So maybe we can take a small number of questions. Again, feel free to use the raise hand button, or else I'll also keep my eye on the chat.
I'm sorry, just to save time, can I ask a question immediately?
Okay, we'll take this one question, sure.
Yes.
Thank you. Saeed, thank you very much, very interesting. Can you please tell this parameter, HA, is it some sort of gating variable which has its own ODE, or is it some sort of constant? It's a little bit too magic for me.
It's a variable. So if I go back to the equation, so that's described by this equation here as
so
HA.
it's a variable, yeah?
So at the beginning it's a variable, but for the slow fast analysis, because it's very slow, it's treated as a parameter. So then it's become constant. So here in this model, it's a variable. But when you come, for example, so here for all of these is a variable, but when you come here to study the underlying dynamics, so it's treated as a parameter. So now it's a constant. So for each one, it remains constant.
Thank you, I got it. And do you have any experimental confirmation that this variable can exist in this range?
Yes. So our parameters, we try to keep them in the biological range. So in the physiological range.
Thanks again, Saeed.
So today I'm going to talking about some analysis of complex bursting patterns in specifically in embryonic respiratory neuron model. And this is a joint work with John Rubin. So I'll begin with a short background about the respiration, which is driven by neural rhythms in the brainstem of mammalian nervous system. And below is a section of the brainstem showing some of the key regions that may play a role in generating respiratory patterns. And today, specifically, I'm going to focus on pre-Basinger, the specific area that is the pre-Basinger complex network of neurons that control inspiration. So these neurons have been known since 1991, and they can produce rhythmic bursting patterns without any sensory input. So there are two possible sources of these bursting. One is the persistent sodium current that activates and only decays very slowly. And also another possible source is the calcium activated non-specific cation current. This is the ICAM, which is activated by the slow intracellular calcium dynamics. So then with these two mechanisms, as we change the two mechanisms, the postnatal prebiotic neurons are able to produce three types of bursting patterns. The first pattern only depends on the persistent sodium current. So we denote that as the M bursting. And the second pattern is only depending on the cane current with the persistent sodium current being blocked. So we call that C bursting. The two mechanisms can also coexist. So we have these M plus C bursting. And we call all these type of bursts that features an active phase of sustained spiking alternating with the silent phase as the plateau bursting. So there is also a special type of bursting emerging in the postnatal pre-burst neurons. That is, it represents a combination of the M burst and M plus C burst. And our previous analysis has indicated that the transition from the M burst to the M plus C burst has to do with the activation of the CAN current. So the idea here is that bursting patterns in postnatal pre-burst neurons are mostly plateau bursting. So then what about the intrinsic bursting patterns of pre-burst neurons at embryonic stage before birth? Do we even have intrinsic bursting? If so, what do they look like? So recently, Cerebellar et al. Provided a first description of bursting activity patterns in embryonic pre-burst neurons, and they also depend on the two currents, persistent sodium current and the CAN current. So there are three different intrinsic bursting patterns here. The first pattern closely resembles the patterns that are expressed in postnatal pre-burst neurons. So we also call that plateau bursting. And this pattern of bursting is mostly sensitive to the persistent sodium current. In the second group, the neurons can produce the new type of bursting that features a sustained depolarization block. So we call that DB bursting. And this bursting is mostly sensitive to the CAN current. And also there is this third type of bursting that is also new, and it represents a combination of the previous two types. So non-surprisingly, this type of bursting is sensitive to both currents. Okay, so with this background, with this kind of data, then I'd like to first try to understand the full range of dynamics encoded within the embryonic pre-burst neurons. And those are the three types. Because the plateau bursting has already been well understood, so I'll just focus on the DB bursting and the mixed bursting. And then also second, we'd like to understand the developmental changes of pre-burst dynamics from the patterns observed embryonically. So mostly these two new patterns. And how does the transition from the two new patterns to those seen after birth, that is the plateau bursting, how do these transitions occur? So there are two possibilities. One is that it might occur some changes of the balance between the two conductances might occur in the single cell level. So that is a CAN-dependent, ICAN-dependent neuron switches to the persistent sodium-dependent neuron. So eventually we only have neurons that are able to produce the plateau bursting. And all these could also occur in network level. So that would be the CAN-dependent neuron vanishes and is replaced by a new population of neurons expressing the persistent sodium. So later on, I'll show you the simulation results and also our analysis. They both support the first possibility. So therefore, in this talk, we will just focus on the first possibility. Okay, so then these are the two goals. And then to do our analysis, we took the model, that's the embryonic pre-burst neuron model proposed by Cerebellar et al., which is also depending on a previously proposed model by Touboul et al. So in this model, we can see that we have voltage dynamics and calcium dynamics. Voltage dynamics is described using the Hodgkin-Huxley. Specifically, I'd like to highlight the two currents that we are interested in, CAN current and persistent sodium current. So the CAN current is activated by the intracellular calcium. Intracellular calcium is denoted by Ca sub I. And in addition to that, we also have the total amount of calcium within the cell, Ca tot, and also the gating variable, IP3 gating variable L. So this is the calcium dynamics. And I won't go into detail of this model. You have seen some of these in previous talks. So then with this model, then what we are going to do is we are going to vary the gating conductances for the two currents. And we find that this model is able to produce different type of bursting, and they are color-coded using this color. Specifically, the model is able to reproduce the three patterns observed experimentally. Okay. So then, as we can see in the yellow region, when we pick the parameters, x-axis is the persistent sodium gating conductance, and y-axis is the gating conductance for the cane current. So when you pick the parameters in the yellow region, we get dB burst. As you decrease Gk a little bit, and also when G and AP is also in some intermediate level, then we get mixed bursting. As we keep reducing the Gk, then we can get plateau burst. And here I'm showing you the plateau burst, which is actually an M burst because we picked Gk to be 0. So it has to be depending on only persistent sodium current. Okay, so these are the three patterns. And using this simulation result, it actually kind of suggests that the change of proportions of these two gating conductance in the single cell model, in this model, actually is sufficient to trigger the transition that we expect. If you kind of decrease Gk or you increase GNAP a little bit, you can get the transition that we wanted. So this is one evidence supporting the first possibility. Okay, so now let's go ahead to explain the dB and mixed bursting. And before doing that, it's well known that the regular burst, such as the plateau bursting, requires at least 2 timescales. And now for the 2 new type of bursting, especially the mixed bursting, it's natural to expect that we might need a 3rd timescale. So we need a method that can apply to more than 2 timescales. And so the method we're going to use is the method that has been used to great success to deal with multiple timescale dynamics, geometric singular perturbation theory. And again, you have already seen this method applied in previous talks. So then start with a 2 timescale system. We rescale time. We can write 2 equivalent systems taking the singular limit. Then we get 2 reduced problems, fast layer problem and slow reduced problem. Slow reduced problem is restricted to f equals to 0, which is the equilibrium of fast layer problem, and it's also called critical manifold. So this method was developed by Neil Fanishaw. And then in the spiking example, this is the time series for the fast variable x. We can then use and the S-shaped curve is the equilibrium, that's the critical manifold. We can use this method to explain the dynamics. Basically, when restricted to f equals to 0, the dynamics is governed by slow reduced problem. That's this problem until passing the saddle node. So f equals to 0 no longer holds. Therefore, fast layer problem takes over, which triggers the fast jump in x, fast variable x, to the upper branch, and then switch into slow reduced problem again. Okay, and then later, John Renzo also pioneered the fast-slow decomposition method to model and analyze bursting dynamics. Here I'm taking one of the fundamental type of bursting, that's the square wave bursting, which is also called plateau bursting as an example. And the reason is that this actually has the same mechanism as the M burst in our model. So then similarly, we have this S-shaped equilibrium, the critical manifold. And in addition to that, we also have the family of periodic orbit branch shown in green, which is stable, born at the half bifurcation. So then after passing the saddle node, trajectory will be attracted by the family of periodic orbit branch and then starts oscillating until passing the homoclinic bifurcation, jumping back to the lower branch. So this is how the square wave bursting is formed. And as I said, if you think of the x as the voltage and replace the y by the slow inactivation variable for persistent sodium current, then this really explains the mechanism for the M burst. And so that is saying M burst is initiated at the saddle node bifurcation and terminated at the homoclinic bifurcation. Okay, so, and then since I said we are dealing with a 3 timescale problem, so what about 3 timescale? So in this paper, we generalized the 2 approaches in the context of GSPT developed for 2 timescale setting to 3 timescale setting. In the first approach, we consider 2 approaches. In the first approach, we make highly use of projections of the critical manifold and superslow manifold, which you probably have seen in Elif's talk. And then in the second approach, we then consider decomposing the system into two coupled systems and then making highly use of the projection of bifurcation diagram and also phase portrait. So in today's talk, I'm going to use the second approach to actually understand the dB burst and the mixed burst. Okay, so start with the dB bursting. To apply the timescale decomposition method, we need to do some non-linearization to figure out the timescales. And this is a rough estimation. We won't be able to get exact timescales for these variables. So this is how we group the variables, and that allows us to do our analysis. And then the idea of the coupled system approach is that we are going to decompose these to two coupled systems. We pick the first one to be the voltage subsystem, and we pick the second to be the calcium subsystem. So now both subsystems, they just involve two timescales. So in this case, voltage and gating variable N are fast, H is super slow. In the calcium subsystem, intracellular calcium is slow, with the other two super slow. So then these two can be understood using the typical fast-slow decomposition method. And then we are going to consider how they interact with each other to get the full dynamics. That is how we get, especially how we get the transition from the fast spiking to the dB phase. Okay, so that's what we are going to do. And so this is the time series for the dB bursting. Calcium subsystem, we use the calcium null surface and L null surface to understand the calcium dynamics. What I plot here is the calcium. This colored surface is the calcium null surface when voltage is fixed at voltage minimum. So then starting at the yellow star, trajectory shown in black will be tracking along the left branch until it's crossing this pink curve, which is hardly seen, but we have a pink curve here. That is the bifurcation curve corresponding to the fold of this calcium null surface when we fix voltage and the two super slow variables CA total and L as fixed parameters. So we get this transition curve. Once trajectory crosses that transition curve at the pink dot, that will trigger the jump of calcium to the right branch, and then it evolves along the right branch until passing the fold, jumping back. So this is the calcium dynamics. And then the voltage dynamics, we can then understand that by taking the super slow variable H as the bifurcation parameter. And here is the voltage. So we also have the projection of the trajectory onto this one parameter bifurcation diagram. So we have, similar to the square wave bursting case that I showed before, we also have an S-shaped curve equilibrium for the fast subsystem. And there is a half bifurcation here at which we have a family of periodic orbit branch born at that bifurcation point. The cyan curve is the H null climb. So we can see the bifurcation structure is very similar to the square wave bursting. And so therefore the fast, this black spiking is also triggered, initiated at the saddle node bifurcation. Okay, but then why it won't be terminated after passing the homoclinic bifurcation, instead it transitions to the dB phase. This is because our calcium, so this bifurcation structure was plotted when calcium is at a minimum. Our calcium, which is the slow variable, is not fixed, is actually changing. So as we change, increase calcium from minimum to the maximum, bifurcation structure is then moving to the left to become the blue, to the blue position. And here I'm showing the half bifurcation and also saddle nodes periodic orbit bifurcation. So this also can be summarized when we take calcium as the second bifurcation parameter. So then we can see as we increase calcium, these bifurcation curves move to the decreasing H direction. So now we can understand how we get the transition from the black burst to a black spiking to the dB burst. So the idea is after the saddle node bifurcation, we already have the spiking. During the spiking, trajectory is moving to the left until at this pink dot, which I explained here, calcium will jump up. There is a little bit delay because calcium is slower than voltage, but there is a jump. And this jump brings the trajectory across the saddle node periodic bifurcation. That is the key reason why it triggers the transition from black spiking to the red dB phase. And to see this more clearly, we can go back to this one parameter bifurcation diagram. As calcium jumps up, now the bifurcation diagram is given by these blue curves. So then the black trajectory is now lying to the right of the saddle node's periodic orbit branch. So therefore, trajectory will be spiral into this attracting part of upper equilibrium branch. So therefore, that triggers the transition from the black spiking to the dB phase. And the dB phase will eventually be terminated at the blue star. That is when decrease of calcium brings trajectory across the half bifurcation. So then the trajectory is now surrounding the unstable part of the equilibrium, upper equilibrium. So it will spiral out and eventually jumping back to the stable equilibrium on the lower branch. So this is the main idea for how we get the dB bursting. And the key idea here I want to highlight is the transition from the black spiking to the red dB is induced by jump up of calcium. If calcium is not jumping, it's not able to jump up, or if it won't cross the saddle node periodic orbit branch bifurcation, then we won't be able to get any dB phase. So that further confirms that dB bursting is depending on the Kane current. And then the mixed bursting case is more complicated. So here I'm showing the time series for mixed bursting and also the two parameter bifurcation that I showed before with the projection of all the three burst cycles using different colors. So we can see that only the red, this is starting from the yellow star, only the red burst is able to cross saddle node and homoclinic bifurcation to form an M burst, which is the square wave burst. But calcium is able to jump up in the other two bursts to cross SMPO curve. And however, the yellow crossing leads to the transition to the dB phase as expected, but the other crossing doesn't give us any dB. So we'd like to figure out what's the difference between the two crosses. Using, just by looking at this figure, they look very similar. So to understand that, we have to go beyond the standard timescale decomposition, and instead we need to consider the competition between the evolving rates of calcium and also the variable H. So then by doing that, we find that the evolving rate of calcium relative to H is slowest during the red burst, fastest during the dB burst, and intermediate during the black burst. So this also quickly explains why calcium is not able to jump up during the red burst, because evolving rate of calcium is too slow. And then to explain why we get these two bursts, we go back to look at the one parameter bifurcation diagram with a projection of the dB burst. So notice that evolving rate of calcium determines how fast the periodic orbit branch is moving to the left. And evolving rate of H determines how fast the trajectory is moving to the left. So in this case, because the dB burst has the fastest movement of calcium relative to H, so the moving periodic orbit branch is able to completely overtake the yellow trajectory. So therefore, the trajectory is able to be attracted by the attracting part of the upper equilibrium. However, this is not the case for the black burst because it only has moderate evolving rate of calcium relative to H. So the moving periodic orbit is not able to completely overtake the black trajectory. Instead, trajectory will track along this moving periodic orbit. That's why we have this change in the spike amplitude. Okay, so to complete the story, we still need to understand why we have different evolving rate of calcium relative to H across the birth cycle, because by our rough estimation shows that calcium is actually a slow variable, H is super slow. But why do we have to still compare them? So that has to do with the levels of the two super slow evolving quantities. And I'll skip the detail for the interest of the time. But the take-home message is that the key aspects arise in periods when separation of timescales is actually different in different regions. So to analyze, we have to go beyond the standard timescale decomposition methods. And also mixed bursting occurs when GNAP and GCAM proportions are intermediate. So that further actually supports the first possibility that changes of the gating conductance proportions actually occur in the single cell level. So then at some point we have a neuron with intermediate gating conductance proportions. So therefore we see mixed bursting in the experiments. So I'll go quickly for the second goal, that's the transition, how change of the two gating conductances leads to the developmental changes that we expect to see. That was seen in the experiments. And we first start with the effect of the decreasing GCAM. And to do that, I fix GNAP at some value, that's 4, and I decrease GCAM. So from top to bottom, GCAM is decreased. And by observation, we can see that we lost the DB feature. Instead, we gain a big plateau. So that is saying DB burst is eventually replaced by the big plateau burst as we decrease GCAM. And also, we don't have any M burst in the beginning for large GCAM, but then later on we gain more and more the M burst. So number of M burst also increases with the reduction of GCAM. And these two qualitative changes are important for us to understand the developmental changes, and they can both be explained using the bifurcation diagram that I show here. But I'll just explain the first phenomena. So basically, these are the time series from left to right, GCAM is decreased. And we can see, we can compare the bifurcation diagrams focusing on the SMPO curve. As we decrease GCAM, this curve is moving to higher calcium level. As a result, it's more and more difficult for trajectory to cross this curve. And once it cannot across the curve, we then lose the DB bursting, as we see here. And instead, we have a big bursting with continuous spiking, no DB phase. So that's why we lost the DB phase. And also, similarly, by considering positions of the saddle nodes and homoclinic bifurcation, how the positions of them change, we can explain why we gain the M burst. And similarly, we can also explain what's the effect of increasing persistent sodium current. So that is, it can promote persistent sodium dependent burst, which is not surprising because it's depending on this current, but it has little effect on the DB burst. So as we can see here, we start with DB burst. As we increase GNAP, we gain some M burst, but we don't lose the DB burst. So that is suggesting developmental change, a developmental increase in the persistent sodium conductance would not in itself be enough to induce the transition that we expect. Instead, we also need some change in the GCAM. So with that, I'd like to conclude. A summary here is that we have linked past analysis of the plateau bursting with the new analysis of DB and mixed plateau DB bursting, and to uncover the mechanisms underlying all bursting dynamics in embryonic pre-box neurons. And specifically, I'd like to highlight that this mixed bursting represents a singular perturbation problem in the non-standard form. And some efforts have begun to explore what happens when individual variables is actually evolving at different timescale in different regions. And our work adds to that literature by providing another example in which not just the individual variable, but the separation of timescales for different variables is actually also in flux. And also our analysis yielded predictions about how changes in the balance between the two bursting mechanisms contribute to the developmental changes of pre-box dynamics. And the idea is that we need a combination of both increase in GNAP and decrease in GCAM. In the single cell level is already sufficient to shift the pre-box neuron activity from patterns observed embryonically to those seen after birth. And so here is a reference, and I'd like to acknowledge my collaborator, John Rubin, again, and thank you all for listening.
Thanks, Yang Yang. Great. So again, if you have questions, I'm trying to monitor the chat and the hands raised thing. And if neither of those work for you and no one else is talking, I guess jump in.
Can I ask a question, or someone ahead of me?
Go for it.
Okay. So thanks for the talk, Yang Yang. As you know, when you talked about the shift in the bifurcation, when you're at low calcium versus high calcium, you have this, the half is shifting to the left.
Mm-hmm.
And you use the argument that differences in how the time constant of calcium relative to the time constant of H
this one
produces the different patterns, which I understand 100%, but the problem is that why would the time constant change? I don't understand that.
So we have, yeah, that's what I skipped here. Because when you non-dimensionalize something, when you get the time constant, it's really a rough estimation, as Krasi said, and also calcium sometimes is slaved to the two super slow variable, if you can see here. So then that's why the time scale of calcium is actually changing.
I see.
And also there are some complex dynamics here that also gives this complexity. Yeah.
Okay. Thanks.
Let's see. Any other questions?
So could I ask a question?
Go ahead, Leon.
Hi, thanks very much. Could you say something about what role these neurons actually play in the generation of breathing, what the current understanding is now, and how these neurons may be playing a role, and what the functional significances of the bifurcations you're discussing?
So in the broader context of the respiratory rhythms, it's actually still not so clear what are the roles of these intrinsic bursting patterns of the pre-bots neurons. So because for the rhythm generation in the network, synaptic coupling also can play a role. And also in our case, we consider the embryonic pre-bots neuron. They actually have done some experiments showing that initially in the early embryonic stage, the pre-bots neuron bursting dynamics play a major role to produce the rhythm, the network rhythm. But then later in the embryonic stage, synaptic coupling starts to also play a role. But I think it's still not so clear what are the role of this one, of the bursting patterns.
Have you discussed this at all with Jack Feldman? Jack is the person who made up the name pre-Botzinger.
I have read his work, but I haven't talked to him in person.
Oh, well, you should talk to him. He's a nice guy, and he actually got a PhD in physics and decided it was better to do experiments than do math.
I will chime in for a second, Leon. I mean, I collaborate and talk quite a bit with Jeff Smith, who's Smith et al. On the 91 paper with the pre-bod. So we're definitely very much in touch with the latest experiments. And I can send you even a couple of other references offline.
I mean, I think in the respiratory system, there's been a lot of question whether it's the oscillations are generated from a network or from pacemakers or bursters. And I know this was a big controversy 30 years ago, and I'm not exactly sure what the situation is at the moment.
Thanks.
Okay, so today I'm going to be talking about stick insect limb segment joint coordination. Before I start, I'd like to acknowledge my advisor, Dr. Jonathan Rubin at the University of Pittsburgh, and our collaborator, Dr. Sylvia Don at the University of Cologne. Okay, so stick bugs, when they're walking, they have two primary gait modes. There's the tripodal, which is three-legged, as you can see here on the left. Each blue leg moves in coordination, and each red leg moves in coordination, and they move sort of in antiphase to each other, which you can see in panel A here. And there's the tetrapodal gait, where we have these diagonal pairs of legs moving in coordination. This is B and C, so the difference between the two is here we have left to right, top to bottom pairing. You can also have left to right, bottom to top pairing. So if you want to coordinate all of these 6 legs, it's going to be kind of complicated. And the first thing you need to be able to do is step.
Could you speak up, please?
So if you want to step, then, or for each step, what you do is you, on this diagram up here, stick your leg forward, put your foot on the ground, drag yourself forward, pick your leg up, move it forward again, and repeat. And that alone can be a fairly complicated process. You have a lot of different moving parts involved. So if you want to be able to step, the first thing you have to do is control and coordinate all of your joints. We are going to be looking at the protractor-retractor joint, the thorax, coxa, hip-like joint. And it's given by this little circuit diagram here. We are only going to be focusing on the CPG units, these two controlling interneurons, and the depressor input beta, just sort of bottom-up sensory feedback from the leg. We don't really care about all of the rest of this because it's the CPG that's going to be driving the action of these interneurons and of these muscles and motor neurons and whatnot. So this came from a paper of Sylvia's back in 2012. I'd like to mention a very, very incomplete list of similar work with this sort of simple joint circuit diagram. Nazare, Srivastava, and Holmes. There's Sparty and Lewis. There's Proctor and Holmes. There's many, many others. So our model equations, fairly standard. We have this voltage equation where we have this ionic action plus an additional input from somewhere. The ionic equations are given by this. I don't think biologically that it's actually a persistent potassium, but it's similar enough that it's convenient notation. So you have a persistent potassium, this leak current, and this gating variable H, which is going to be slow. So this is a non-spiking model, which is interesting because the actual insect neurons are non-spiking. Then we have these input currents, which you can sort of follow from the circuit diagram. I'm not going to do an equation dump. So we have input from beta onto interneuron 4. Interneuron 4 drives C1 and IN3. IN3 inhibits C2. C1 and C2 are mutually inhibitory, and they each have a separate top-down influence. So since this is, I should mention also, each of these synapses is very, very steep. So the drive or inhibition that each cell receives from its synapses is going to be sort of an all or nothing type of thing. Now, since we're in a fast, slow system, we can view this with, we can analyze this with moving null lines. And then because we have these steep synapses, so the beta input that IN4 gets is going to be this heaviside function composed with the sine. We have this tau here, that's the period of the gate. Big gamma is just a phase shift to let us decide where in the beta period we want drive to begin. And we have this little gamma, which is going to give us our duty cycle. And I should point out that from here on, I'm only going to be talking about the tetrapodal gait pattern. Okay, so moving null lines. What happens when beta is off and we start somewhere over here? Then since this is a fast, slow system, we have to immediately rush to our slow null line. And once we hit that, we slowly crawl along it toward our fixed point. And if that was the end of the story, we would just stay there forever, and that would be very boring. But we have this Heaviside function, so eventually this is going to turn on. And when it does, we find ourselves in the beta on point, hanging out over here by nothing. And again, we're in a fast, slow system, so we have to rush over to our voltage null line. And then we, again, crawl along it until we hit this knee. So we're trying to go down, but we've hit this knee. There's nowhere further down to go. So we have to jump across to the other branch of this cubic and then follow toward our fixed point. And again, if that was the end of the story, we would stay there forever. But because we're oscillating, at some point beta turns back on. So we have to rush back to this one and start the whole process again. And this is going to be the driver of most of the activity in the circuit. So if we want the interneurons to be entrained, then we need a knee clearance condition and a stability condition. So let's talk about the knee clearance. So if we start at H0 here, on the driven null line at the beginning of the drive, we follow our green null line down until drive ends. We jump across to our undriven null line, and then we follow it back up. And what we require is after one period tau, the point where H0 is equal to H of tau is above this knee. Because now tau is over, beta is going to turn back on, and that leaves us clear to jump back to the active side of our null line. I should mention this vertical dashed line here is the synaptic threshold. And so when that fails, I'm sure you can guess what's going to happen. We start at H0 when drive begins. We crawl down, drive ends. We crawl up. And now, when beta comes back on, we rush over toward driven null line, but the closest point on that is below threshold. It's below its knee, so we're going to get stuck here. We only have the one stable fixed point, so we stay here for the next entire tau period. So that is non-trained. We can't get entrained if this happens. So I said we needed knee clearance and stability. So let's talk about stability. So it can happen that the orbit where H of 0 equals H of tau, this brown one here, is unstable. And so if that's the case, then on every other tau cycle, you're going to drift either alternatingly above and below that orbit. And then eventually you can see you're going to just barely clip that knee. But even though you've just barely clipped it, that's still not good enough. You're stuck, you have to drift down, and you stay off for another tau period, and so you fail to be entrained again. On the left here are different parameter regions for different periods tau and drive strengths G at 4. You can see if it's low enough, this knee will be above this fixed point, and there's never any hope of entrainment, so that's great, we don't have to check there. Otherwise, we have this red curve where we fail to clear the knee, and we have this blue bar where we've lost stability, and stranger things can happen. So we have a baseline state where the CPG is entrained. And for CPG entrainment, we require a little bit more than we do for the interneurons. Since they're mutually coupled, we also require that for each tau period, each CPG unit has exactly one active and one inactive phase. And we also require that they are never simultaneously active. It's okay for them to be simultaneously quiescent, but not simultaneously active. And the reason for that is each CPG unit is controlling a different muscle, protractor or retractor. And if they're both quiescent, well, that's fine, nothing is moving. But if they're both active, it's trying to attract and retract at the same time. That's probably not going to work. It sounds painful, and we're not going to even try. Okay, so in4 is driving C1, and in3 is inhibiting C2. So in each cell, these green and red null lines are sort of switched. And the green null line is
Quick reminder, if you're not the speaker, please make sure you're muted. Marcello, you're muted now.
We good?
Yep.
Hello?
You're good.
Right, so the interneurons are having complementary effects on the two CPG units. So the green and the red null lines are flipped. And you can see this complementary nature on these voltage traces in the bottom left. Here, purple is interneuron 4 and blue is C1. So you can see that C1 follows the interneuron activity fairly closely. And red is C2, which is sort of antagonistic to the interneurons and to C1. Or oppositional, at least. So I want to do a brief aside on escape and release. So this is upside down from our setup. You can see the sigmoid is going from low to high, while ours go from high to low. So it's upside down, but mechanistically it's the same. So let's see how this works. So we have two neurons, an inhibited neuron and an active neuron. This is in the escape regime. The active neuron crawls along this upper null line, and it's trying to keep the inhibited cell off as much as it can. It has this stable fixed point here, so it will never turn off unless it's forced to. Meanwhile, the inhibited cell, it's trying to break out. It's like it's in jail and it's tunneling its way to freedom. It crawls down and it hits this lower knee here. When it does that, it wants to jump to this branch of the lower null line. But before it does that, it crosses threshold. And now that it's crossed threshold, this active null line, or this active cell, is now being inhibited, so it has to rush over to the lower null line. And now that this cell has crossed threshold, this lower cell no longer sees this lower null line and goes straight to the active branch of the upper one. And then this oscillates. And release is very similar. We start with an inactive cell and an active cell. This time, our inactive cell crawls down, but it can't ever get to that point of freedom. So if the last one was sort of like somebody escaping from prison, this is more like somebody just serving their time. The inactive cell crawls up, reaches this knee and says, okay, you can go, I'm going to go to retire, or go to bed or something. Crosses thresholds. The inactive cell now rushes to the upper nucleus. The active cell no longer sees the upper nucleus and goes straight to the inactive or the lower nucleus. And again, this oscillates. So the difference between these two mechanisms is that here it is the inactive cell that's driving the oscillation, and here it is the active cell that's driving the oscillation. So in the baseline case, the CPG is neither. Without any input from beta, C1 will always be quiescent, C2 will always be tonically active, and it will stay that way forever. But by changing the synaptic strength from the bottom up signals and increasing the top-down inputs, we can perturb these into a more escape-release type setup. And sometimes this can cause problems. So if we drive C1 enough, it can instigate a release type mechanism. So let's walk through that really quickly. So at point 1, drive begins. We jump over to our driven null cline and crawl down until point 2, here, where we hit one of our knees. And so we jump over to this inactive side of our driven null cline, and we don't actually make it all the way to the red null cline or anything intermediate because C2 is still being inhibited from C3. And that inhibition is strong enough to prevent its release when C1 tries to release it, when C1 stops inhibiting it. So that means that C1 stays on its driven null cline up until point 3, where it hits another knee. And this time, it jumps up across threshold again. And so what we have here is that in a single tau period, C1 is undergoing 2 quiescent and 2 active phases. It's a sort of a weird twitch in the muscle, which is not good. This is not what we want, so this is not entrained. But we can recover entrainment with top-down modulation. So again, point 1, beta begins. We crawl down to point 2 and we jump down. And what happened last time is that, well, the inhibition from C3 is preventing C2 from becoming active. But now if we increase the top-down drive, we have this intermediate null cline where C2 is being inhibited from C3, but not by C1. And with enough top-down drive, this intermediate knee is going to move through a fold, and this fixed point will be destroyed. And now C2 is free to become active at point 3. And now that C2 is active, C1 is being driven by N1, but inhibited by C2, so it has its own intermediate null cline, which it will stick to until point 4, where drive ends. And you can see here, there should be a threshold there. We have 1 active and 1 inactive phase per cell. And perhaps more interestingly is that we've pretty dramatically altered the duty cycle of the CPG relative to that of beta. And likewise, well, if we can increase top-down to C2 so that it can escape when it's partially inhibited, maybe we can do that when it's fully inhibited, and sure enough, we can. And again, this can cause problems. So following the same schematic, at point 1, drive begins, C1 turns on, C2 turns off, and C2 crawls up its inhibited null cline until point 2, where it's free to escape. And at that point, C1 is being inhibited from C2, but driven from ion 4. So it has this orange intermediate null cline here. And when it collides with that null cline, that is above threshold. So C1 stays active, so C2 stays on its driven, or its inhibited nullcline, but is also active. And eventually, C1 will fall off of a knee and become quiet, and then everything will be fine from there. But we have this region where we have simultaneous activity. And again, that's trying to protract and retract at the same time. It's not a good idea. This is not entrained. But again, we can recover entrainment. So what we've done here is reduce the drive to C1. And I mean, it does two things, but primarily what it does is that it raises the knee of this intermediate nullcline. So what happens now? Drive begins, C1 turns on, C2 turns off, C2 escapes, and C1 is able to just make it under that knee. And since it's able to make it just under that knee, it's able to get all the way through to below threshold so that they are no longer simultaneously active. Slightly interesting note is the timing. So C2 stays on its fully inhibited nullcline for a couple of milliseconds because it has to wait for C1 to actually reach threshold here. And again, you can see that we're entrained, we don't have simultaneous activity, but we've made some pretty dramatic changes to the duty cycle of the CPG relative to that of beta. In fact, here it is nearly fully reversed. I know this is more of a curiosity than anything else. This is not escape or release. It's a matter of the timing. So recall N4 receives the beta input. It forwards it to C1 and N3, and C2 is the last cell to know what's going on. So if C1 is driven very excessively, so we're past the release portion, the nullclines are now monotonic, so really there's no knees to fall off of. What can happen is, this is hard to see, but as drive ends here, then C2 hasn't realized that the drive has ended yet, right? It gets it one step behind C1 because it has to go through N3 first. So C1 knows that drive has ended and it tries to inactivate, and it has this intermediate nullcline where it is being inhibited by C2, but it is not being driven by N4. And when it reaches this intermediate nullcline, it is just above threshold. And since it's just above threshold, it continues to inhibit C2. And so if C1 has strong enough inhibition, even though C2 is not being inhibited by N3 anymore, it can force C2 to stay quiescent. And so we have this little gap where C2 fails to activate, and this is not entrained. And weirdly enough, we can recover entrainment by driving C1 even more. So we require bottom-up drive for this case, because the idea is to increase the differential between the driven and undriven state. So what's happening here is this brown nullcline is what we had previously. And now we're going to increase the bottom-up drive. My screen just died. Well, I'll try to find it. We're going to increase the bottom-up drive and move the green nullcline to the left. This is going to increase the voltage for the H infinity curve, and we're going to travel down this null line slightly faster than before. And now that we're traveling down it slightly faster, well, when beta turns off, when we run into that intermediate null line, now we are just below threshold. And since we're just below threshold, we freeze C2 and entrainment is allowed to continue. I think that's my conclusion slide.
Yeah, you see the conclusions.
Okay, so we're able to analyze each cell as a sort of planar fast slow system, and we can analyze the different drives using this moving null line analysis. So using this approach, we can see where entrainment is possible or for what parameters entrainment is possible. And where entrainment fails in the CPG, we can use top-down modulation to shift these null lines in such a way that we can recover entrainment. And more than that, we can use this to alter the duty cycle of the CPG, which could provide some insight into gate transitions.
Okay, thanks, Marcella.